These are the germ cells or gametes, and when they join together during fertilization, there will be zygote formation. Nucleic acids, these are polymers. Poly means many, so they are many, made up of the nucleotides. So polymers of phosphorylated nucleosides. That is the building blocks of DNA and RNA. The building blocks of DNA and RNA are nucleotides. When you say nucleosides, nucleosides different from nucleotide. Nucleosides are made up of sugar and nitrogenous bases, okay, without phosphate. When we add phosphate, phosphorylation or phosphorylated means adding phosphate. When we add phosphate on the nucleoside, we get nucleotides. So, nucleotide, an organic molecule that is the basic structural unit of DNA and RNA. DNA and RNA are the genetic material in eukaryotes and bacteria too. Nucleotide means nucleoside plus phosphate. This is the three parts of nucleotide, and the nucleotide consists of nitrogenous bases, the three components. Nitrogenous bases, 5 carbon sugar molecule, and phosphate group. In DNA, the nitrogenous bases are adenine, guanine, thymine, and cytosine. In RNA, uracil replaces thymine. There is no uracil in DNA and there is no thymine in RNA. Nucleoside is the building blocks of RNA and DNA. Nucleoside means without phosphate. When we add phosphate, we get nucleotide. These are compounds consisting of a purine bases such as adenine or guanine, or pyrimidine bases such as thymine or cytosine attached to ribose in RNA or deoxyribose in DNA. These are the sugars, which pentose sugar or 5 carbon sugars found in the nucleic acids. Ribose in RNA, ribonucleic acid, deoxyribose in DNA. They attach it on the first carbon of the pentose sugar, that is 1' carbon. Here is the three parts of the nucleotide: the phosphate part, the sugar part, and the nitrogenous bases. We say nitrogenous bases because they contain nitrogen. Okay, this is a nitrogen base, that is adenine. 1' carbon, there are 5 carbons in a pentose sugar. This is a pentose sugar. Carbon 1, carbon 2, carbon 3, carbon 4, and 5. Therefore, the nitrogenous bases will be attached on the first carbon or 1' carbon. This is nucleoside, only sugar and nitrogenous bases, no phosphate in the nucleoside. These are the bases. Purine base structurally, adenine look like this, guanine is this, and the pyrimidine base, thymine, cytosine, uracil. These are the double ring, 1 ring, 2 ring. Double rings, the purine bases are double ring bases, and the pyrimidine bases are single ring bases. Then DNA, deoxyribonucleic acid, the abbreviation is that. A complex organic molecule that is capable of self-replication. This is very interesting. The DNA molecule can self-replicate, or self-replication means it can duplicate itself. And it carries and stores genetic information. DNA is a material of heredity and is found in all non-living organisms. Except some viruses, RNA viruses, almost all organisms' genetic material is DNA. DNA, the molecule encodes the genes that allow genetic information to be passed to offspring or generation. DNA sequence, the ordered sequence of nucleotides on a strand of DNA. Example, TAA, GT, and etc. What this indicates? These are nitrogenous bases: thymine, adenine, guanine, thymine, cytosine, thymine, guanine. And this, when we represent by T, we are saying this is one nucleotide. A, the other nucleotide. Again, A, the other nucleotide. G, the other nucleotide. When they make bond, they will make the DNA. So, in every nucleotide, the two components are the same. Sugar, here, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. The sugar and phosphate are the same throughout every nucleotide. What makes one nucleotide from the other differ is the bases. Here is thymine, here is adenine, then adenine, guanine, thymine. Therefore, nucleotide 1 differ from nucleotide 2 by differing of the nitrogenous bases. Among the three components of a nucleotide, such as phosphate, sugar, and nitrogenous bases, the two components, phosphate and sugar, are the same. The nitrogenous bases makes different. They are vary from one nucleotide to the other.